The LitmusMilk test. The LitmusMilk test is a simple broth inoculation. Using aseptic technique, you will inoculate 4 different litmus milks. Prior to inoculation, you will need to label each tube with the name of the bacteria with which it will be inoculated. When inoculating the litmus milk, it is helpful to use a larger than normal amount of bacteria. The heavy inoculation will help to jumpstart the reactions. The litmus milk often takes several days to over a week for some reactions to be able to be read, so using a heavier than average inoculation will help speed up those reactions. If after 24 to 48 hours of incubation reactions are still not observed in some of the tubes, they may need to be left for up to 7 or 10 days. Remember to use aseptic technique when transferring the bacteria. When inoculating with a large amount of bacteria, you may need to scrape the loop onto the side of the tube to remove all of the bacteria that's on the loop. Be careful not to insert too much of the loop into the tube. This could introduce contamination into the litmus milk. Repeat this inoculation for the next 3 tubes. Litmus milk is used for the cultivation of bacteria in the dairy industry. It is also used for the differentiation and identification of many gram-negative and gram-positive organisms. Litmus milk consists of skim milk and the pH indicator litmus. Litmus will turn pink in acidic conditions and blue in alkaline conditions. In addition to indicating pH changes, litmus will also indicate if redox, reduction-oxidation reactions have taken place. Litmus milk reactions are sometimes difficult to determine, so proper length of incubation is important. Now shown is an acid reaction in litmus milk. In an acid reaction, the milk sugar lactose has been fermented. Fermentation gives off acid waste products which causes the pH indicator litmus to turn a bright pink. Clots are also sometimes seen in acid reactions, in addition to reduction. Shown here is an acid reaction with reduction in the bottom of the tube. Reduced litmus will turn white. This is also a semi-solid, which is a clot, and if gas production had occurred, fissures and cracks would be seen in the semi-solid clot. Fermentation with a clot in litmus milk and fissures and cracks or gas production is known as stormy fermentation. Now shown is an alkaline reaction. On the left is a control or uninoculated litmus milk. On the right is an alkaline reaction. In alkaline reactions, partial breakdown of proteins has occurred. This results in the release of ammonia and other alkaline byproducts. The pH indicator litmus turns a dark blue in the presence of alkaline conditions. In this tube, no reduction has occurred. Now shown is true reduction. In true reduction, some protein catabolism or breakdown has taken place. This explains the blue band at the top of the tube. However, litmus has taken part in redox reactions and has been reduced. Reduced litmus will turn white. During these reactions, the reduced litmus and leftover milk proteins oftentimes become a semi-solid or curd in the bottom of the tube. There will not be gas production in alkaline or reduction reactions. Now finally shown is digestion. Digestion is the complete breakdown of all proteins in litmus milk. This is also known as peptonization. Since all the proteins in the milk were broken down, it is no longer cloudy, and peptonization releases alkaline conditions or waste products. This causes the litmus that's in the media to still turn blue. The presence of whey and a blue band at the top indicates digestion, also known as peptonization. Now shown are the 4 most common litmus milk reactions. Starting from the left, they are acid or fermentation, alkaline, digestion, and finally reduction. Remember, reduction can occur in both acidic and alkaline conditions. Shown here, starting from left to right, is the control, followed by alkaline, alkaline reduction, digestion, acid reduction, and acid. The litmus milk test can be used for the identification of both gram-negative enterics and gram-positive rods. It is often used to differentiate between Bacillus anthracis, the causative agent of anthrax, and other Bacillus species. In addition to bacterial identification, it is sometimes used for the cultivation of different lactobacillus species used in the dairy industry for the making of cheese, yogurt, and other dairy products.